So let's continue. We were looking at the issue between Mr. and Mrs. Smith. I said the two of them are heterozygous for widow's peak, but their union produces a child that is not or that does not have this widow's peak. So Mr. Smith accused his wife of infidelity. Is it justified or not? Okay. Now, you can't just by looking at the question tell either it's justified or not, but you have to either do a test course, either do a normal course rather, or you use Punnett square to get it. You need to know the genotype of the offspring that will result from this union. So look at it here. Since Mr. Smith is heterozygous and his wife too is heterozygous, so these are their genotypes. So let's look at it. Are they going to have a child that does not have widow's peak? This child and this guy will give us homozygous dominant. This child and this guy will give us what? Heterozygous dominant, same as parent. Then this guy and this guy will also give us heterozygous dominant just like the parent too. And this child and this child will give us what? Homozygous recessive. So this is the child that is causing the issue. So the problem is not with Mrs. Smith, and it's not even with Mr. Smith. But the point is, Mr. Smith does not know that he's carrying the trait. And also his wife too is carrying it. So by the time the two traits came together, it gave rise or birth to this kind of child that does not have this widow's peak. So Mrs. Smith is not what? She's not infidel in any way. And Mr. Smith is not, cannot be justified for that accusation. They both caused it. Okay, then the next question. Please, I want you to pay attention to this question. We have not dealt with this part of biology in the part I've explained, but it is very, very common in JAMB question. Okay, a man with genotype OO, please stay with me. I'm sure many people will always run away with courses that is involving blood. A man with genotype OA married a woman whose genotype is OB. Then what are the genotypes and phenotypes of the entire family? Now please stay with me. Genotypes. Please stay with me. The man is OA, the woman is OB. Right? Now you would agree with me that for this man to be OA, as his phenotype rather will be A, and that of the woman will be B. Right? This is the phenotypes. These are the phenotypes rather, and these are the genotypes. Okay, so let's do the cross to get what will now be the genotype of the children. OA and OB. Let's see. This guy and this guy. So this child is going to be O. The blood group is going to be O. Let me quickly give you this. I hope you know that the best blood group is O. And it's not O, it's not just ordinary O. It is O negative. Please stay with me. It is O negative. We won't go into that. Now, then for this guy, it is OB. For this guy, no, this guy, it is OA. And for this guy, it is AB. Okay, now let's look at it. These are the genotypes. Let's look at the phenotypes now. This child is O. This child is B, this one. This O here now does not mean anything. And this one here is A, and this one is AB. Is that clear now? Is that clear, please? Stay with me, please. Most times, it may not be written like this. They can use what we call immunoglobulin O, immunoglobulin B. They can use what we call immunoglobulin O, immunoglobulin A. This guy is B, this guy is O. I don't know if I'm going too deep. Okay, but for clarity's sake, just take this like this. Like this OO is O, OB is B, OA is A, then AB, you have to write the two of them. Are you getting me now? Now, this blood group A, the reason why, this blood group O, the reason why it is not taken as significant is because it is O. Let me give you an instance. When you have pure water and somebody is bringing gutter water, someone is bringing dirty water, If you pour the dirty water inside the pure water, it becomes a problem. But if you pour the pure water inside the gutter water, it does no harm. So if you are having O and A, this is pure, this is the impurity here. It does no harm. Are you getting my point now? But when you join this dirty one, A and B together, you can't neglect any of them. Because they both have their own consequences. That's the reason why I can't leave any one of them. But look at this one now. I can afford to leave out the O because it is pure. It doesn't cause any harm. That is why it is pure. It is called blood group O. O means what? It doesn't have A, doesn't have B. It is pure. It does not even have razors. I'm not going into that. Okay, so let's move on. Okay, let's look at the possible question that can come out in JAMB from the topic that we just treated. The change in genetic makeup of an organism resulting in a new inheritable characteristics. What am I talking about? What change now can happen in the gene of an organism? Let me show you something. Please stay with me. Okay, now please look at this. This is DNA, right? This is DNA, and this picture here is showing DNA directionality. What do I mean by that? It is DNA doesn't lie head to head, just like sexual reproduction in earthworm. They lie head to tail. Okay, now the two strands run anti-parallel. You have 5' to 3', then you have 3' to 5'. You don't have 5', 5', 3', 3'. This is what we use to label head tail in DNA. 5', 3'. So please stay with me. If you have G here, that G will come in. I want to explain something to you so that before we go back to that question. Now look at this. There are some basic components of DNA. We have what we call sugar. It has sugar. What type of sugar do we have in DNA? It has deoxyribose sugar. That is the reason why it is called deoxyribose. The sugar has lost adversity, which is why it is deoxyribose. It has a phosphate backbone. Look at it. It says sugar phosphate backbone. This is it. This is the phosphate backbone. It has nitrogenous bases. DNA has 4 different bases inside it. We have what we call adenine. We have thymine. We have cytosine. And we have guanine. Please stay with me. Please. When scientists are talking about genetic engineering, what they do basically is to look at everything that you have in your DNA. Look at this. You know, the point is, if you take this T away and you replace it with something else, it's going to affect the structure. If you take this thing away, like you remove this C, which is cytosine. If you remove the C, obviously it might affect this G, because this G cannot pair with T, it can only pair with C. Look at it. Can you ever see C and G, G and T pairing together? It doesn't happen because they are different. Adenine can only pair with thymine. You can only have A to T. You can have C to G. You can have T to A. You can have G to C, but you cannot have T to G. It doesn't work. It's error. Are you getting me now? So if you take out this C, right, it can affect the entire structure. And when the entire structure is affected, the DNA is affected. When the DNA is affected, the owner, which is the organism too, is affected. Okay, so let's go back. Now, so let's go back and look at the question. It says the change in the genetic makeup of an organism resulting in a new inheritable characteristic. That means in a new characters that we can inherit. It's known as what? It is called mutation. All of us know mutation. When an organism has undergone mutation, a lot can happen to the organism. It can change. It can be good. It can be bad. Right? But please, I hope you know that there are some food that we eat that are mutagenic. Let's keep that. So the answer for this is mutation. Now the next one. The organic molecules present in the nucleus are, you have DNA, you have RNA, protein and DNA, We have RNA, we have ATP and RNA. Please, the answer is A. DNA, not C. DNA. Now, the point is this. When you talk about nucleus, I want to go into something. Please, let's quickly look at this. The DNA is found in the nucleus, right? RNA does not occur in the nucleus. Are you getting my point now? RNA does not occur in the nucleus, even though DNA is the carrier of the information that you need to make protein, which you now use to form who you are. But DNA is in the nucleus, RNA is not in the nucleus. So DNA will look for how to transfer the information inside it from the nucleus into the RNA that is outside. Take for instance, you want to leave, you are in school, you are in your school now, right? You are in the school and you want to leave the school premises, but you cannot, they can't give you access. You can't go out of the gate because it is not yet closing time. Then you have your what? Let's say you have your principal beside you. He will help move you out. Are you getting? He will help move you out so that you can go to wherever you're going. So this DNA cannot go out of the nucleus, but RNA cannot come out of the nucleus too. Are you getting me now? So you look for a way to change the information inside it into the format of the RNA. Please, I won't go into that for now. Then let's look at what's the next question. Okay. The next question says, albinism is an expression of traits caused by recessive traits. Albinism is caused by recessive trait, not dominant trait. Now the next one. The male sex cells, also called gametes, are produced by a process called spermatogenesis. C. The male sex cell called gametes are produced by spermatogenesis, and the female sex cell is produced through a process called oogenesis. Okay, then the next one, number 9. Which of the following describes an example of fertilization in higher organisms? Which of these describe an example of fertilization in higher organisms? A. Fusion of egg and sperm nuclei. B. Pollen grains landing on a sticky stigma. C. Ejaculation of the egg and the pollen grain. This cannot work. Egg and pollen grain, it doesn't work. Then D. Ejaculation of the egg into the female body. The answer is D. The answer is D. Okay. Then the next one is, what is the phenotypic ratio of a dihybrid cross between two heterozygotes? Now, please stay with me. In this class, we have not done the hybrid cross exclusively, and I won't do it because of time. But there's one thing you want to know. I am very sure that JAMB will not give you anything aside this. JAMB will not ask you for the genotypic ratio of the hybrid. They won't ask you because the answer is not regular, and it can be annoyingly long. But if it is phenotypic, which I am 100% sure they will ask, the answer is 9331. Now, what are they talking about? Remember, I told you about monohybrid and dihybrid. Please stay with me. I told you about monohybrid and dihybrid. So, in the case of dihybrid, please, I gave you something like this, right? So, if you want, the other time we did something like this. We did this kind of cross, right, to get the offspring for this. So if you are to do that for this, you're going to have 16 offspring that will result from this cross. You're going to have 16 offspring, and if you are to analyze their phenotypes now, you're going to have the ratio 9:3:1. JAMB will not go below that. I'm very sure. Okay, so let's move on. The next topic is cell division, and I want you to prepare your mind, get your books ready. If you want to drink water, you can quickly go. When we come back, we continue with cell division.